Thank you, Dr. Vasu, and the seminar organizing team for giving me this opportunity to showcase some of my research activities with you. So I'm an assistant professor at the Department of Civil, Environmental, and Construction Engineering. First, I would like to start with a brief bio here. I did my master's in the Netherlands, where I was part of a large project looking into the emerging pollutants, micropollutants, which was funded by European Commission. Then I moved to Toronto, Canada, to pursue my Ph.D. in civil engineering at the University of Toronto, which I completed in 2014. And then I worked as a postdoctoral researcher there for about a year until I joined UCF civil and environmental engineering department in 2015. While my broader training and interest is advanced water and wastewater treatment, particularly interested in membrane processes to treat water and wastewater and for water reclamation, focusing on removing contaminants of emerging concern, or CECs. Why I'm interested in this is because they pose risks to the aquatic species, various aquatic species, and human health. I have contributed to the interaction of various classes of CECs with their interactions with water matrix components and membrane processes that would ultimately that may impact the removal of this contaminant using membrane processes. And very recently, I have been actually researching on the fate and transport of CEC in water as well. At UCF, I teach undergraduate courses like environmental engineering operations and processes, and chemical process control. The graduate courses, currently I'm teaching industrial waste treatment this semester and water treatment. Now, here is the big picture of today's topic. Membrane processes do a good job in treating water and removing contaminants depending on their configurations and separation mechanisms. But why I'm focusing on the contaminants of emerging concern, as I mentioned earlier, they pose risks. They've shown adverse consequences to various aquatic species, which has driven considerable attention regarding their impact on human health. Now, conventional membrane treatment that are used, they show variable rejection of the contaminants of emerging concern. Now, what do I mean by the contaminants of emerging concern? They are these various classes of contaminants, pharmaceuticals, endocrine disrupting compounds, personal care products, and so on. These various classes that have not are not typically monitored in the environment, but they have the potential to cause adverse ecological and human health effects. Now, these contaminants, these chemicals might be in our waters for a long period, but probably were not recognized because until sophisticated analytical tools to detect them were developed. Now, the conventional membrane processes show variable rejection of the CECs, and sometimes this removal are very compound specific. Other key challenge associated with membrane processes is that they get clogged, which we call membrane fouling, which will decrease the service time and cost money. Now, I have investigated the interactions of during my past research and current research, investigated the the selected groups of CECs with water matrix components and membrane processes to see how these interactions influence their removal when using membrane processes. And in our membrane process lab at UCF, now we have been actually investigating or looking for so-called all-rounder membranes so that they can actually address all this or overcome this shortcomings that I summarized. Now, membrane, the commercially available membranes can be modified or functionalized using nanoparticles, nanotubes, or we can fabricate mixed matrix or thin film nanocomposite membranes. We can use carbon-based materials to improve the performance of the membranes. But how these membranes interact with the CECs need to be studied, because so that we can actually efficiently design membrane processes targeting those contaminants of emerging concern. And also that is the reason I have been working on improving the performance of membranes, because most of the previous studies have shown when they use this, followed this modification, attempted this modification, they showed compromised solute rejection in exchange of the volume of water produced, and vice versa. So I'd like to improve the performance. That's what we're investigating, to improve the performance in terms of rejecting the CECs or removing the CECs, but not at the cost of volume of water produced. And also we want to improve the resistance to deposition of fouling materials on the membrane surfaces. So with my past experience with a range of CECs, including pharmaceutically active compounds, endocrine disrupting compounds, and so on, we recently tested this group of contaminants called PFASs, perfluoroalkyl and polyfluoroalkyl substances. The PFASs, it's the so-called forever chemicals, because they're very persistent in the environment. So we tested their removal using a conventional, first started with the conventional nanofiltration membrane to see how it removes PFASs from different source water that are used to produce drinking water. And specifically, we investigated the most widely investigated PFASs, that is perfluorooctanoic acid and perfluorooctane sulfonic acid, using a bend scale setup. And an analytical method was developed, and we used our in-house liquid chromatography tandem mass spectrometry to detect this, measure these chemicals. Now, what we observed was that the PFASs were removed in higher percentages in the natural waters compared to the lab prepared water, which is likely due to the presence of the water matrix components, the natural organic matter, the colloidal and the cations in the groundwater. There were significantly higher natural organic matter compared to and calcium ions specifically compared to the laboratory prepared water, which we also demonstrated by correlating the removal with the presence of the calcium, magnesium ions, and the presence of organic matter in the water. Then we took a low pressure membrane, which is a ultrafiltration membrane, because it will now require significantly less energy. That means less cost. So we modified this membrane by coating it with oppositely charged polyelectrolytes, polyacrylic acid, specifically polyacrylic acid and polyallylamine hydrochloride, and then immobilized silver phosphate nanoparticles within the coatings. And it resulted in increased hydrophilicity of the membrane. The membrane became more hydrophilic, and also the membrane surface charge became more negative, which that's what we want when it comes to improve the performance in resisting the foulant deposition. Here is a plot of permeate flux. That's the water produced as at different stages of the filtration. The blue line represents the unmodified membrane, and the green is the modified functionalized membrane. As you can see, during the fouling stage, they follow the similar trend, but when we clean the membrane, the unmodified membrane continued to decline in flux, but the modified, the functionalist membrane, maintained its performance. And also, the functionalization imparted antimicrobial property to the membrane, as you can see from this plate counts. This is for the modified membranes, and these are for the different dilutions of for the unmodified membranes. You can see there is less attachment of microorganisms. Otherwise they would just start partying there and colonize, and eventually lead to biofouling of the membrane. Now we are determining more important properties due to the functionalization, and then testing the removal of these classes of emerging contaminants, including PFOS and PFOA. This would have been completed by now if we hadn't been affected by the pandemic. We'll use this bench scale setup and analyze this contaminant using our LC-MSMs. Now here is another example of membrane functionalization. This time we are functionalizing a forward osmosis membrane, and this was funded by, it was a student design competition one, awarded by EPA P3 program. In this case, we modified a forward osmosis membrane by coating it with polyelectrolytes and embedding nanovalent zero valent iron within the coatings. The overall goal was to demonstrate a feasible approach whereby we can actually reclaim water from one impaired quality source, for example, storm water, from using another impaired quality source, for example, a reverse osmosis treatment plant's rejected stream, which would otherwise have to be, managed properly before it is disposed. And we modified this membrane, and we actually found out that once the membrane is uniformly coated, we actually tried different bilayer coatings, and once the membrane is uniformly coating, starting from this point, you'll see that the flux actually did not reduce, was not declined, but a little bit actually improved. That's what is good, because at this stage the membrane was able to remove very efficiently nutrients and selected heavy metals, as well as PFOS and PFOA. Here I show you EDX mapping on the surfaces. As we increase the bilayer coatings, you see more fluorine elements, indicating that it was able to absorb more PFOS and PFOA. This project was funded by Hinckley Center for Solid and Hazardous Waste Management. In this case, we used electrospun fiber mats, applied it on ultrafiltration membranes to see if it improves its efficiency in removing heavy metals from landfill leachate. And actually, we found out that it significantly improved, that just the fiber mat itself significantly improved the removal of this heavy metals from different. First, we tried with different water matrices, and then we applied this mat on the. You can see at this cross section of this SEM cross section. We applied this mat on the ultrafiltration membrane, and it actually improved. And we tested actually real leachate matrix, and we saw significantly improved removal of heavy metals from landfill leachate, and we think that this is likely because of metals association with the functional groups available in the rich functional groups available in the polyelectrolytes. Now, the PFAS removal was not actually within the scope of the study, but we tested, and we actually found out that 40% of PFAS was removed by just those fibers, but not much PFOA. But we we didn't have enough funding to really continue these studies, but in future we're going to continue with these studies. Now I'll continue with my search for all-rounder membranes, but in addition I'll investigate CEC removal using other treatment techniques. Recently funded a project by FDEP that is led by Dr. Naibing Chang. We are investigating removal of nutrients
out of time
from using biological adsorption media, and my long-term plan is to quantify the interactions between the CECs and membranes so that we can predict the removal and removal mechanisms using membranes. And finally, I would like to thank my lab members. There's a list of distinguished collaborators from UCF and outside UCF. I would like to also acknowledge funding agencies. Last but not least, thanks to my department chair and faculty members and office staff who are continuously supporting us in our endeavor. Thank you for your patient hearing.